This is chapter 17. We're going to talk about the reproductive system. Our learning objectives today are to differentiate between meiosis and mitosis. We'll describe the process of spermatogenesis and oogenesis. We're going to describe the structure of spermatozoa and list the components of the male reproductive system and describe the functions of each. We will talk about the functions of the accessory structures of the male reproductive system and the components of the female reproductive system, and again, describe those functions of each component. We'll describe the events that occur during the Ovarian Cycle and then the stages of the Estrus cycle and describe what events occur during each stage. We'll be able to differentiate between monestrus, diestrus, polyestrus, and seasonal polyestrus-estrus cycle intervals. So let's start with chromosomes, very basic. Coiled masses of DNA in the nuclei of the cell. Each cell in an animal's body, except spermatozoa or ova, contains identical chromosomes, and they have a diploid chromosome number. This diploid number is the total number of chromosomes in the nucleus of each body cell, and it's all the same for each of these cells within each species, except for those reproductive cells. So the spermatozoa and the ova have half that number. It's always an even number. Chromosomes always occur in pairs. When there is an odd number, that's when we have a genetic abnormality. Sex chromosomes, they're designated as either X chromosomes or Y chromosomes. If both the sex chromosomes are X, then the chromosomes are XX, the individual is genetically female. Now, I want to make a note here. We are talking about mammals, okay? Mammals have XX and XY. When we go to reptiles, they're going to be a little bit different. If one of these chromosomes is X and the other one is Y, then the individual is genetically a male. So this is how it looks, X and a little tiny Y. Here's an X chromosome and an X chromosome. Here's what it looks like under an electron microscope. All these little fuzzy parts are the coils of the DNA with little side chains coming off of them. And these are the blueprints for everything that happens within the body. The haploid chromosome number is made by reduction division. It's the total number of chromosomes in each of the daughter cells is reduced to half the number of the parent cell. So here's diploid, we got 2 of each, and haploid, we have 1 of each. So this ensures that the fertilized ovum from the union of the spermatozoa and the ova has a diploid number. So 2 plus 2, or 1 plus 1 equals 2. The haploid chromosome number in the reproductive cells results from meiosis. So here's the difference between meiosis and mitosis. Mitosis is the way that most body cells divide after the cell first makes a duplicate copy of its chromosomes. So it makes a duplicate copy, and then half the chromosomes go to one daughter cell and half go to the other. The genetic makeup of the 2 daughter cells is exactly the same as each other and as the parent cell. So it makes a copy of itself and sends half of that copy to one daughter and half of the copy to the other daughter. Meiosis is when the chromosomes do not first produce a duplicate copy of themselves before the daughter cells divide. So in mitosis, we have a copy. So we have 2 times the original number of chromosomes, so 4 altogether. So then we divide by 2 daughter cells, we get those 2 chromosomes to one, 2 to the other. And in meiosis, we don't make that copy first. So we just have those 2 chromosomes and split them in half. So one goes one way, one goes the other. Half of the total chromosomes, 1 from each diploid chromosome pair and 1 sex chromosome, go to each daughter cell. Which chromosomes go to which daughter cell is entirely random. And that's why if you have brothers and sisters, you may look different from your brothers and sisters, or you have different characteristics from your brothers and sisters, because these within meiosis, we have a random separation of those chromosomes. So here's mitosis again. We have 2N. This is all body cells have 2N paired chromosomes. They make a copy of themselves, that becomes 4N, and then they divide 2N. In meiosis, we have 2N. That goes to 4N, that goes to 2N. So it starts with mitosis and then completely splits off. Okay, and these are little sperm cells. Spermatogenesis, this is the production of male sex cells, and this occurs in the seminiferous tubules of the testes. So you should know where spermatogenesis occurs. Where do we make sperm cells? In the seminiferous tubules of the testes. They're produced continuously and in very large numbers. So it starts with a primary spermatocyte, which is 2NXY. We split this 2NXY into 1NX, 1NY. We have then this splits again, copies itself, splits again. We have an X, a Y, a Y. These are the spermatids. When they grow tails, they become spermatozoa. Primary spermatocyte divides by meiosis into secondary spermatocytes, and you have your haploid number. Then the secondary spermatocytes divide by mitosis into 4 spermatids. Those spermatids grow tails and undergo other physical changes that convert them to spermatozoa. When the spermatozoa are fully developed, they detach and are carried to the epididymis for storage before ejaculation. Half of the spermatozoa produced have an X sex chromosome, and half have a Y sex chromosome. So you have a 50-50 chance of having a girl or a boy, and you've heard before that the male determines the sex, and that's because they just happen to have Y opportunities with their spermatozoa. For oogenesis, the development of the egg, or the production of the female sex cells, this occurs in the ovarian follicles. Now, female has a fixed number of primary oocytes at or soon after birth. So they don't continuously make them. When they're born or soon after that, they have as many ovum eggs as they're ever going to have. Oogenesis produces small numbers of ova at a time. So we have the fixed number of oocytes, and then going through an estrus cycle produces the ova. Here's our primary oocyte. This starts as a 2N or XX for a female. We split into half, and one of these halves, the secondary oocyte, one is much bigger than the other. Cytoplasm-wise, has same number of chromosomes within it. Now, these are polar bodies. Polar bodies are the support cells for this big ovum. And this one will then split by mitosis into 2 different cells, and this one will split by mitosis into this is 2 different same-size cells. This is 1 large cell and 1 small cell. So primary oocyte divides by meiosis into a large secondary oocyte and a small polar body, and they each have that haploid chromosome number. And then the secondary oocyte then divides by mitosis into the ovum and 3 polar bodies. Within the male reproductive system, we have things that produce male sex hormones that help to develop the spermatozoa and deliver the spermatozoa to the female site at the appropriate time. So the testes produce sperm in the seminiferous tubules, store them in the epididymis, and they produce hormones. The testes are located outside the abdomen in the inguinal region, or in the perineal region when we're talking about cats. They're housed in a sac of skin called the scrotum. The spermatozoa structure, we have a head which contains the nucleus, or the powerhouse of the cell, and all of those chromosomes that are so important. It is covered by an acrosome. Think of this acrosome as the hair, but it's hair with digestive enzymes. So the acrosome contains digestive enzymes that help the spermatozoan reach and penetrate the ovum. The digestive enzymes help to enter the ovum. The mid piece is the large concentration of mitochondria, and here's where actually the energy is, and it's arranged in a spiral pattern, and that spiral pattern helps to move that tail, whip it back and forth. So the tail has muscle-like contractile fibrils, which produce a whip-like movement of the tail and propel the cell forward when it's normal. It's not always normal. All right, so the development of the testes. There is a gubernaculum, which is a band of connective tissue that attaches the testes to the scrotum. The testes start up by the kidneys where the ovaries are and gradually are pulled caudally and ventrally. They come through the inguinal ring, which is a small break in the tissue of the abdominal muscle. It's an opening in the muscle through which the testes descend into the scrotum. The scrotum is a sac of skin that houses the testes, and it helps to regulate the temperature of the testes because the testes must be kept slightly cooler than body temperature. There is a muscle, it's called the cremaster muscle, and it passes down through the inguinal ring and attaches to the scrotum. It adjusts the position of the testes relative to the body depending on temperature. So here we have animals that have descended testicles, and they are all the way down in the scrotum or mostly down in the scrotum. And then we have can pull it up. It can be pulled up into the more close to the body wall so that the testes warm up. So it's basically thermoregulation. Spermatic cords, these are blood and lymphatic vessels, nerves, and the vas deferens. So these are all connected to the testes. They form a pampiniform plexus. This is this pampiniform plexus, which is a meshwork of veins that surround the testicular artery. This helps cool the temperature of the testes. It maintains that testes at slightly lower than body temperature, and then it actually warms blood back to the body temperature before it returns to the abdomen. Tunics, okay, tunics are shirts, coverings, okay, and in this case they are connective tissue that form a sheath-like layer around the testes and the spermatic cord. It helps to protect it. There's the visceral vaginal tunic, which is a very thin inner layer. Anything that says visceral means that it covers the organ closely. The parietal vaginal tunic is on the outside. It's the thicker outer layer, and it forms that fibrous sac around each testis and spermatic cord. And then there's the tunica albuginea. This is a white tissue. It's a fibrous connective tissue capsule surrounding each testis beneath those tunics. So here is the testicular anatomy. Here is the testis itself. Here is the epididymis, and the seminiferous tubules go into the epididymis, and the epididymis from the head to the tail of the epididymis turns into the vas deferens, which carries the sperm to the female. Tightly connected onto the testes is the tunica albuginea, and then we have our tunica, our visceral tunic, and our parietal tunic. Right, within the seminiferous tubules, we have spermatogenesis occurring. It's a long convoluted U-shaped tube which is attached at both ends to a system of ducts called the rete testis. There are interstitial cells which are in between the seminiferous tubules, and these are endocrine cells that produce androgens like testosterone. Androgen hormones. Sertoli cells are cells within the tubules that support developing spermatids. So I think of Sertoli cells as support cells. They're kind of like the polar body of the ovum. They do support, and when I think of Sertoli cells, I think of a big Italian bodyguard supporting the little tiny spermatids. So I don't know if you want to think of it that way as well, but it helps me to remember Sertoli. The duct system. After detaching from the Sertoli cells, spermatozoa enter the rete testes. Then they flow through the efferent ducts. Efferent goes away to the epididymis. The epididymis is a single long convoluted tube that connects the efferent ducts of the testes with the vas deferens. It contains the spermatozoa, so it stores them and it helps them to mature. So it's kind of like where they go to college. All right, the epididymis has a head region. It's the site where spermatozoa enter from different ducts. It has a body region, which lies along the surface of the testis. There's the tail region, which continues on as the vas deferens. So look at this. These tubules are just winding roads, okay? And it's this rete testes that contains the seminiferous tubules, and there are septa or separations between different portions. Vas deferens, or the ductus deferens, is part of the spermatic cord. It passes through the inguinal ring and then separates from the spermatic cord and connects with the urethra. There's the ampulla, which is an enlargement of the vas deferens just before it joins the urethra, which is found in some species. Those contain glands that also contribute material to the semen. The vas deferens has thick, smooth muscle walls, which function to propel sperm quickly from the epididymis to the urethra at the time of ejaculation. So the vas deferens comes up from both testes and goes into the urethra. There are seminal vesicles, the prostate, the bulbourethral gland. These are glands that contribute to the semen. So within the urethra, we have the pelvic portion, which is the entry point of the vas deferens and the accessory reproductive glands. That is the seminal vesicle, the prostate, and the bulbourethral gland. We have the penile portion, which runs down the length of the penis. The spermatozoa from the vas deferens and secretions from the accessory reproductive glands enter the urethra and are pumped out as semen. This carries, the urethra also carries urine from the urinary bladder outside the body, but urine flow is temporarily blocked when ejaculation occurs. You have to remember that urine is at a certain pH that will actually kill sperm. Accessory reproductive glands. Ducts of all of these accessory reproductive glands enter the pelvic portion of the urethra, and different species have a different combination of accessory reproductive glands. They produce an alkaline fluid that helps counteract or buffer the acidity of the female reproductive tract, keep the sperm alive. The prostate gland is one of these accessory reproductive glands. It surrounds the urethra, has multiple ducts that carry secretions into the urethra. We have the bulbourethral glands. They're also called Cowper's glands. These are ducts that enter the urethra near the caudal border of the pelvis, and they secrete a mucinous mucus fluid just before ejaculation that helps to clear and lubricate the urethra so that the sperm can flow right through. The penis is composed of muscle, erectile tissue, and connective tissue. It has a large blood supply and many sensory nerve endings. There are three main parts of the penis: the roots, the body, and the glands. The roots of the penis are bands of connective tissue called crura that attach the penis to the brim of the pelvis. The body of the penis are two bundles of erectile tissue, the corpus cavernosum urethra and the corpus cavernosum penis. They are fibrous connective tissue and blood-filled sinuses, and when the sinuses engorge with blood, that inelastic connective tissue around the sinus generates hydraulic pressure, which causes the erection. The glands of the penis is that distal end. It has numerous sensory nerves. And then we have the prepuce, which is the sheath of skin that encloses the penis when it is not erect. The inner portion of this prepuce is smooth and has a moist mucous membrane so that the penis can move within it without getting damaged. For the canines have an os penis. It is a bone within the penis, and the urethra runs right through the groove on the ventral surface of this bone. Canine penis has a bulb of the glands, which is an enlargement toward the rear of the glands, and it can engorge with blood. You may notice it on your male dogs, even if they are not, or if they are neutered. It will engorge with blood, and it will reach full size after ejaculation. What this does is it helps the penis to remain clamped in place by contraction of the muscles surrounding the vagina and the vulva. So dogs, when they mate, become locked in place, and they will turn backwards to each other so that their rear ends are facing, after he mounts and ejaculates for 15 to 20 minutes. And after that, then the erection of the bulb will subside. And that helps to keep the semen within the bitch so that there's a greater chance that she will become pregnant. In other mammals, there is something called a sigmoid flexure. This is an S shape of a non-erect penis of the bull, the ram, and the boar. And this has a higher proportion of connective tissue to erectile tissue than other species. Erection results from straightening of the sigmoid flexure from internal hydraulic pressure. This causes the penis to protrude from the prepuce for breeding. So this sigmoid flexure is normally there when the animal is going about its normal business. When it is erect, it straightens out, which makes this penis much longer so it can penetrate further into the female. There's a picture of the penis with the retractor penis muscles, the sigmoid flexure, and the glands at the distal end. Reproductive functions, erection, results from the parasympathetic reflex triggered by sexual stimuli. So again, it's parasympathetic, so nothing fight or flight or stress can be occurring or should be occurring during this parasympathetic reflex. This often involves olfactory cues and behavioral changes. So they will be able to detect when a female is ready to be bred. Arteries will dilate and increase blood flow into the penis. Veins are compressed against the brim of the pelvis by contractions of muscles in the roots of the penis, and so it keeps blood within the penis. Ejaculation is the reflex expulsion of semen. Semen moves from the accessory reproductive glands into the pelvic portion of the urethra, and rhythmic contractions of the urethra pump the semen out into the female reproductive tract. Here we are at the female reproductive tract. Okay, so we have within the reproductive system, we have several ligaments that we'll talk about: the ovaries, oviducts, uterus, the cervix, the vagina, and the vulva. The ligaments that we see, especially when we're doing a spay, we have the broad ligaments, which are sheets of peritoneum. It's part of the peritoneal covering in the abdomen. These help to suspend the ovaries, oviducts, and uterus within the abdomen. We have a mesoovarium near the ovary, mesosalpinx, and the mesometrium. Salpinx is near the where the ovum travels into the uterus, and the mesometrium is the uterus. These contain blood vessels and nerves which help to feed the tissue. Now, this is a litter-bearing animal, and litter-bearing animals have two sides of the uterus. They have a right uterine horn and a left uterine horn, and then they come together in the body of the uterus. The suspensory ligament of the ovary is what holds the ovary close to the kidney. It's the ovarian end of that broad ligament, and it's attached to the body wall in the area of the last rib. There's also the round ligament of the uterus. It's you don't see it here, but it's over here a little bit. It is fibrous tissue and smooth muscle in the lateral fold of the broad ligament on each side, and often we need to break down that round ligament as well. We break down the suspensory ligament of the ovary in order to pull the ovary outside of the body wall so that we can clamp the ovarian blood vessels and tie those off when we're removing the ovary and the uterus. Ovaries are in the dorsal abdomen near the kidneys, and that's where the testes start as well. There's a species variation in appearance. It is the site of oogenesis, and it helps produce estrogens and progestins, which you should remember from the intro class. Here's the ovarian cycle, and we've talked about this, and I like to go over it again and again because it's really important. It's the development, the cycle is the development of the ovum, it's ovulation, the formation of the corpus luteum after ovulation, and then degeneration of unripened follicles in the corpus luteum if we do not have a pregnancy or after a pregnancy. Ovulation is influenced by follicle stimulating hormone first, and then by luteinizing hormone. So first we have follicle stimulating hormone that stimulates the generation of the follicle to feed that oocyte, to grow that follicle, grow to continue to feed that ovum and help it to mature. And then it ruptures because of luteinizing hormone. And then after it ruptures, we get a corpus hemorrhagicum. It fills with blood, and then it turns into the corpus luteum, which will produce progesterone in order to support pregnancy. So in ovarian cycle, within a uniparous species, this is one mature ovum produced per cycle. This is the horse, the cow, and the human, unless we have twins or triplets being born. And then we'll have usually more than one ovum produced per cycle. With horses, twins are not desirable because often one of the twins will be much smaller and much weaker, and they will actually cause problems with the other twin, and we typically don't get twin births. It's very unusual to get twin births in horses. With multiparous species, we have multiple ova produced per cycle. That's why we have litters: the cat, the dog, the sow, the rabbit, the ferret. The ferret's not as much. They usually have twins or triplets. Ovarian cycle. We have the primordial or primary follicle, which is an immature oocyte surrounded by a single layer of follicular cells. We get follicular recruitment or follicular activation. That's when follicle growth is triggered. It is stimulated to do this by follicle stimulating hormone, FSH. The follicular cells will then thicken and multiply into multiple layers. We call those granulosa cells. The follicle grows rapidly as the granulosa cells multiply. Granulosa cells produce increasing amounts of estrogen as the follicle becomes larger. And the fluid-filled spaces that form between the granulosa cells, these spaces gradually merge into one large fluid-filled space called the antrum. A mature follicle is called a Graafian follicle or a vesicular ovarian follicle. This is mature when the production of estrogen peaks. And remember, estrogen feeds back to the hypothalamus to slow down the production of follicle-stimulating hormone and to increase the production of luteinizing hormone. The oocyte is on top of that granulosa cell mound. It's called the cumulus oophorus. Cumulus is a kind of a fluffy cloud, and it's surrounded by a thin layer of granulosa cells called the corona radiata. So this thin layer is kind of like the crown of that oocyte. All right, so with ovulation, this is actually seeing an egg ovulate from a follicle. Here's the ovary, here's a enlarged follicle, here's the surgical instrument, and here's the egg coming out. Ovulation is the actual rupture of that mature follicle and the release of the reproductive cell into the oviduct. The surface of the mature follicle will weaken and rupture. Fluid is released from the antrum along with the ovum, and it's still surrounded by that corona radiata. Look at that beautiful golden crown. And the empty follicle fills with blood, and that's called the corpus hemorrhagicum. Ovulation occurs spontaneously in most species as a result of rising levels of LH, luteinizing hormone. Luteinizing hormone breaks open that follicle. There are induced ovulators where ovulation can only occur after breeding, after copulation with a male, and that's in the cat, the rabbit, and the ferret. Now, ovulation still occurs because of LH release, but LH release and an increase, a surge of luteinizing hormone only occurs after copulation in these animals. Corpus luteum. This is formed by divisions of those granulosa cells that line that blood-filled follicle. It's now blood-filled because it's ruptured and we get blood in there, and it's corpus hemorrhagicum. It's influenced by the continued stimulation of luteinizing hormone, and it starts to produce progestins, primarily progesterone. This is necessary for maintenance of pregnancy. Progesterone protects pregnancy. There's an endocrine signal to the ovary that causes the corpus luteum to be maintained only if the ovum implants in the uterus. And the ovum is going to implant in the uterus if it is fertilized. Oviducts are also known as fallopian tubes and uterine tubes. They extend from the tips of the uterine horns, and they are the infundibulum, the end of it, is an enlarged opening, kind of like a funnel, at the ovarian end of each oviduct. Fimbriae are muscular projections that form margins of infundibulum. They help to properly position that infundibulum. So oviducts have smooth muscle fibers in the walls with ciliated cells in the lining, and there are muscle contractions and cilia movements that guide that ovum toward the uterus. And that's where the oviducts is actually where fertilization usually occurs. Now, it is really amazing that this egg here, this is an egg that is being ovulated into the abdomen. And somehow, in this oviduct, we have this infundibulum that reaches out and grabs it. The fimbriae help it, reaches out and it grabs that egg in the right, it's at the right place at the right time, grabs that egg, and that egg travels through the oviduct into the uterus. And it's in that oviduct where fertilization actually occurs. So sperm has to get all the way through the uterus, all the way up to the oviduct in order for fertilization. So it's a pretty amazing thing that happens. Now, if that egg, which is ovulated into the abdomen, does not make it into that fallopian tube, then we will get an ectopic pregnancy, or we can get an ectopic pregnancy, and that can be a problem. So these oviducts are pretty, they're pretty small. They're at the end of the uterine horn, and here is the uterus, okay? And this is a litter-bearing animal, so we have uterine horns and then a body of the uterus. The uterus is a hollow muscular organ. Usually it's Y-shaped, Y-shaped, especially Y-shaped within litter-bearing animals. The uterine body forms the base of the Y. The uterine horns form the arms. Uterine wall layers. There is the endometrium. Endo means the inside. It's the lining of the uterus. It's composed of simple columnar epithelium and simple tubular glands. The glands secrete mucus and other substances. Then we have the myometrium, which are thick layers of smooth muscle. Then we have the perimetrium, which is the outermost layer covered by the visceral layer of the peritoneum. At the bottom of the cervix, the bottom of the uterus, we have the cervix, which is a smooth muscle sphincter between the body of the uterus and the vagina. It controls access to the lumen of the uterus from the vagina. It's normally tightly closed except during estrus and parturition. So it opens for estrus when the female is most receptive to a male so that sperm can enter. And it opens for parturition so the fetus can leave. The vagina is a muscular tube that extends caudally from the cervix and connects it with the vulva. The vulva is composed of the vestibule, the clitoris, and the labia. The labia are the lips on either side. The vestibule is the opening in the middle, and the clitoris is within, and it's just above the urethra, typically. The urethra opens on the floor of the vestibule. So if we were trying to go into the urethra in order to catheterize an animal, we'd have to go down here to the bladder instead of up through the cervix. Up through the cervix, we're doing artificial insemination. Here's the clitoris. It's before the urethra. Estrus cycle intervals. Polyestrus are animals that cycle continuously through the year if they are not pregnant. That would be cattle and swine and humans. Seasonally polyestrus are animals that have seasonal variations in the estrus cycle. Horse, long day breeders. Sheep are short day breeders. Cats are more long day breeders as well. Diestrus are animals with 2 cycles per year, and that usually occurs in the spring and the fall, and that's a dog. And monoestrus are animals with only 1 cycle per year, fox and mink. The estrus cycle is the time, and you can see it's spelled with just a U. It is a time from the beginning of one heat period, estrus, to the next. I'm sorry, estrus cycle actually should be spelled with an OU. See here, estrus cycle. When we're talking about points within the cycle, it is just the U. When we're talking about describing the cycle as an adjective, it's an O. Proestrus, estrus, medestrus, diestrus, and anestrus in some species. Species that are polyestrus do not go through an anestrus cycle. Proestrus is when follicles begin developing and growing. We have an output of estrogen that increases accordingly, okay? The linings of the oviduct, the uterus, and the vagina thicken. The vaginal epithelium begins to cornify and forms a layer of keratin on its surface. Estrus is that period of sexual receptivity in the female. The estrogen level production peaks. Ovulation occurs near the end of estrus in some species. So it'll change a little bit between species. If you're breeding, you need to know where ovulation is going to occur. Induced ovulator species, for like the cat and the rabbit, remain in a prolonged state of estrus if they are not bred. Medestrus is the period during which corpus luteum develops. The progesterone produced by the corpus luteum temporarily inhibits follicular development in the ovary. So we're not going to get any more follicular development because we have some progesterone in the corpus luteum. The lining of the uterus is prepared for the implantation of that fertilized ovum, and the cornified epithelial lining that developed in the vagina during proestrus and estrus is lost. So when we have a dog that we're trying to breed, and we do a vaginal swab and we see cornified cells, we know that we are in proestrus or estrus. As soon as we lose those cells, we know we're in medestrus. And that means that the corpus luteum, the ovulation has occurred, the ovum is likely in the oviduct, and the corpus luteum is getting for pregnancy. So it's during those 3 to 6 days that we want to get her bred. Diestrus is when the corpus luteum is at maximum size and exerting maximum effect. If the fertilized ovum implants, so it should have been fertilized by now, then the corpus luteum is retained well into pregnancy. If no pregnancy occurs, then the corpus luteum degenerates at the end of diestrus, and then the animal either goes back into proestrus or the ovary shuts down and the animal goes into anestrus. Diestrus is the time at which we see pyometra occur. Anestrus is the period of temporary ovarian inactivity that's seen in seasonally polyestrus, diestrus, and monoestrus animals. The ovary temporarily shuts down. So for these animals, if we don't get them bred during this certain period, then we're going to have to wait until next time. Here's what I want you to go back and review. Anything you have questions on, please write those down and bring them to class.